An internal medicine doctor working in a hospital is called into the emergency department to see a 20-year-old with severe pain in his back and left thigh. The physician learns that the patient has sickle cell disease and experiences painful episodes several times each year. Usually, the patient manages episodic pain at home. But in this case, the pain needed emergency assessment and relief. The patient receives fluids and pain medications, and the internal medicine doctor admits him for observation and tests. Sickle cell disease is a lifelong and debilitating genetic condition. As recently as 2014, people with sickle cell disease still died more than 10 years earlier than people who didn't have the disease. If conditions like sickle cell disease are so fatal, why do they still exist today? And what does a little pest called the mosquito have to do with all of this? My name is Florence Yuen, and this is Darwin MD. Mosquitoes are the villain in many diseases, but few realize their role in sickle cell disease. The disease isn't triggered by a bug bite, but research shows without the insect, sickle cell and many other genetic conditions probably wouldn't exist. What's the link between mosquitoes and sickle cell disease? See agriculture for that. When humans began cultivating plants and animals for food, they needed a consistent supply of water for crops and livestock, especially if they lived in drier areas that didn't get a lot of rain. Irrigation solved that problem, but triggered another. Standing water is perfect for disease-carrying insects, like mosquitoes. As mosquitoes proliferated, so did a parasite they carried, Plasmodium. These parasites are natural weapons against humans. They invade red blood cells and cause them to explode, leading to yellowing of the skin, liver damage, kidney failure, and even death. This infection is otherwise known as malaria. Despite billions of dollars and decades of eradication efforts, malaria still infects millions of people and kills almost 500,000 each year. Imagine facing malaria without such technological advances. Prehistoric humans needed to confront the pesky Plasmodium parasite. How did they do it? Thankfully, human genetic variation provided many solutions to this problem. Spontaneous genetic mutations led to random changes in the structure of different humans' red blood cells. Some of these changes could prevent malaria infection while still allowing red blood cells to do their job of carrying oxygen and nutrients throughout the body. People with these mutations were more likely to survive and pass them on to their children, meaning that these mutations became more common as generations passed. Some of these mutations got rid of certain proteins on the surface of red blood cells that the Plasmodium parasite would usually bind to, such as the Duffy antigen. This meant that the parasite couldn't invade and damage red blood cells because it couldn't attach to them. Others changed the structure of certain protective proteins, such as glucose 6-phosphate dehydrogenase, meaning that infected red blood cells were more likely to be destroyed before the Plasmodium parasite could spread to other cells. The mutation that most attracts our attention is one in hemoglobin A, the protein responsible for binding to oxygen in the lungs and carrying it to the rest of the body. This particular mutation changes just one of the amino acids that make up the hemoglobin A protein, causing the protein to take on a different structure. This is a Superman mutation, known appropriately as hemoglobin S. When red blood cells with this mutation don't get enough oxygen, the hemoglobin S inside starts to stick together, forming long chains that stretch red blood cells from their usual flexible disc-like form into a rigid crescent shape. These sickle-shaped cells can't bend and squeeze into small blood vessels like normal red blood cells can. However, having red blood cells that can become sickle-shaped offers a remarkable level of protection against malaria. Studies in many sub-Saharan African countries have found that hemoglobin S offers as much as 90% protection against malaria. This is the sickle cell trait. In short, that means the sickle cell trait emerged as an evolutionary adaptation against mosquito-borne infections such as malaria. That still doesn't explain the persistence of hemoglobin S over millennia. People with sickle-shaped cells are much less likely to die from malaria, but they will perish instead from sickle cell disease. How can humans win? Sickle cell disease and other related diseases still exist today because of an evolutionary concept known as heterozygote advantage. Remember that every gene in our bodies consists of two alleles, one from each of our parents. A gene is homozygous if the two alleles are the same, and heterozygous if the two alleles are different. If a person is homozygous for the sickle cell trait, this means that they have two copies of the mutated allele that produces hemoglobin S. They have some protection from malaria, but they also can't make any normal hemoglobin A, so they are susceptible to the pain and complications of sickle cell disease. If a person is heterozygous for the sickle cell trait, though, they have one copy of the hemoglobin S allele and one copy of the hemoglobin A allele. Because they have some hemoglobin S, they receive protection from malaria, but they still make normal hemoglobin, so they don't develop sickle cell disease. They get the best of both worlds. In essence, when dealing with sickle cell trait and other genetic mutations influenced by malaria, we are playing the genetic lottery. If your parents have sickle cell trait and live in an area where malaria is common, they are more likely than not to survive long enough to give birth to you. In turn, there is a 50% chance that you will be like them and gain the benefits of both hemoglobin A and hemoglobin S. However, there is a 25% chance that you will be born with two copies of the hemoglobin S allele and have to confront sickle cell disease. That is why debilitating conditions like sickle cell disease still plague us today. The risk-benefit analysis came out favoring sickle cell trait. In exchange for a 25% chance of having a painful genetic disease, we got a 50% chance of increased protection from a common and deadly infection. Turns out that genetic conditions like this didn't only evolve in response to malaria. Another possible example involves a mutation in the protein CFTR. The protein produces a watery fluid in the lungs and intestines that flushes microbes away. When this protein isn't made properly, it results in a disease we know as cystic fibrosis, where people produce thick, dry mucus that clogs the lungs and gut. That makes breathing more difficult and blocks food digestion. Cystic fibrosis may be another example of traits that stay in the gene pool because of heterozygote advantage. In 17th to 19th century Europe, diarrheal diseases like typhoid fever and cholera proliferated due to poor sanitation. These bacteria produce toxins that attach to intestinal cells. Cholera in particular made a toxin that kicked the CFTR protein into overdrive. The overstimulated CFTR protein pushed liters of fluid out of the intestines every hour in an effort to get rid of bacteria. If people suffering from cholera didn't get enough fluid replacement, they could die from dehydration within a few hours. The relationship between cholera and cystic fibrosis is still under investigation. However, the hypothesis is that the cystic fibrosis mutation in CFTR limits the severe fluid losses caused by the cholera toxin. A person who is heterozygous for the cystic fibrosis mutation would lose half as much fluid as someone who didn't have the mutation. That mutation might have been the key to neutralizing cholera infections and limiting deaths from dehydration. Genetically inherited conditions like sickle cell disease and cystic fibrosis show us that humans' relationship with their environment is like many relationships. Complicated. We can't separate our genetic composition from the environment in which we develop. Each influences the other. These adaptations go back to the key evolutionary concept of trade-offs. Every genetic change comes with risks and benefits, and evolutionary principles help determine what changes are beneficial enough to persist across generations. When patients like the one we saw at the beginning of this episode walk into the hospital, they unconsciously carry all of this genetic and evolutionary history with them. Exploring this history can help us understand the context behind their condition. Maybe it holds the key to finding treatments too in the future, as evolutionary medicine, like the process of evolution itself, presents new challenges and opportunities for the field of medicine. Thanks for watching Darwin MD.